Good day everyone, welcome to the course on biological data analysis and visualization with R. So, the agenda for this week is to do the WGCNA analysis, right, which is weighted gene co-expression network analysis, and in the last class I have just introduced about WGCNA, what WGCNA means, some of the applications of WGCNA and very briefly the steps. So, the agenda for this class is to do and look at the R package WGCNA, and then we'll take a data set and we'll actually start analyzing that data set. So, this is what we'll do in this class. So, we'll first look into the WGCNA package in R, right, and then we'll look into the gene expression data that we can work with. I will describe the data, and then of course we'll download the data and start the hands-on process. We'll set up the process, and then we'll follow this over subsequent classes, because this is a very long process. Okay, so here is the reference again to the WGCNA package, and I'll show you in a moment where you can find this package, and what we have a manual for this package, the functions that you can use. And the data that we will be using is this data from NCBI GEO. The data set ID is this GAC number is given, right, and this data set is from this paper. Again, the reference to this paper is given. So, please feel free to go and read this paper for more details. So, what this data contains, right, and what this paper talks about is, so it contains gene expression data for diffuse type gastric cancer samples. So, it also has other type of cancer samples, right, intestinal gastric cancer samples, as well as the expression data for some normal samples. Okay. So, we will look into that data. This is a really vast data set, but we will take only a few of them and we will do the hands-on, because otherwise it will become very very intensive, and it might take a lot more time. So, for the data we need to get the metadata, right. So, we have to have the data preparation. So, we also have the metadata which will describe, right, what you have in there. Okay. And we also need to know the gene annotation, right, which gene ID, gene number, etc. Okay. So, let us first look into the R package, and then we will look into the data. Okay. So, let us move on to the R repository, right, and we will start with there. Okay, here. So, this is the package WGCNA, right. This is part of R repository, not Bioconductor, and this is the paper, right. So, this is actually described in this paper here. The R package I have given the reference, and here is the reference manual for this. Okay, package WGCNA. Okay, and you have the functions, right, all these functions that are available to us. Okay. So, we will use some of these functions from this package. For example, we use the as I can see, for example, we will use the network function, which might be here. Yeah, network function is there, right. So, we will also have the soft threshold function somewhere, right. Let us see, where is it? Okay. So, we have this soft connectivity, then we have Tom plot, Tom similarity, all these things. So, all these functions are there, and then of course, as for other packages, you have all the details and also with some examples. Okay. So, again, please feel free to go through this documentation and find out the functions that could be useful. Okay. So, this is the package. Now, let us move on to the data. So, the data set is the GAC 113255. Okay. So, this is a data set, right, from diffuse type gastric cancer. Organism is Homo sapiens. This is the description, and this is a data set for 2020, and this is expression profiling by high throughput sequencing. This means this comes from next generation sequencing. So, again this is the summary of that paper and then the overall design. So, this is something that is also important for us to understand before we actually jump into the analysis, right. So, here is the RNA sequencing data of 140 tissues. These include diffuse type gastric cancer, 107 samples. There are intestinal type gastric cancer tissue, 23 samples, and normal gastric tissue n equals to 10 samples. Okay. And this is the protocol, how this was generated, how the data was done. So, the sequencing was performed in paired and reads and NextSeq 500 platform in Illumina. Okay. So, this is next generation sequencing. These are the contributors, and this is the paper where this is described, okay, the molecular signatures of prognostic subtypes. Okay. So, here is the data, and then this is the platform Illumina NextSeq 500 Homo sapiens, and you have the description of these samples. Okay. So, here is the RNA sequencing, this is the name of these samples. So, this is the GSM number. So, each number refers to a single sample, right. So, here is a single sample, it is a sample type SRA, this is short read, this is the raw data, and this is the cancer type, which is the intestinal cancer type, intestinal gastric cancer. Then you have the extraction protocol, the data processing protocol, how the data was processed, right, how the data was mapped and the read counts were generated, etc. So, all these things are mentioned. The genome build that was used is HG38, this is the human genome version. So, all these things are given, and here is a log2 counts per million data. Okay. So, again we have discussed this in our earlier course, what this million counts per million, how do you report this data. So, this is a count data, which means how many reads roughly are mapping to each gene in this data set. Okay. So, what we have this data is we have downloaded this data, and we have this file, right. So, let us say what we have is the, I will show you the data, but then we also need something called the metadata from this. Okay. So, metadata means we have to see these are the sample names, which sample belongs to tumor, which samples belong to normal. So, this is something easy to see. For example, you see the letter T here. So, these samples they belong to tumor patients. Okay. So, these are the tumor tissue, and N means, right, you can see these N ones, these are normal usually. Okay. But then you have some samples where you do not have any T, N, etc. So, those are difficult to understand, right, what those are. So, we need metadata which will tell us, right, which sample is which type, right, whether it is a cancer tissue or a normal tissue. Okay. For that we have the series matrix files, and here I will just click on it, and you will see we can simply download this series matrix file, and we will have this, we can pre-process format, and we will get this information, the metadata that we need, right, which sample belongs to which type of tissue, whether it is a normal tissue or cancer tissue. Okay. So, we will see that in a moment. We have downloaded all of this, and I have pre-processed them, and we have generated this, we have this information already. Okay. Okay. So, let us now look into the data. Okay. As I said, here is the data, you can see. Okay. So, we have downloaded this raw count file. Okay. So, this raw count file, so maybe I will open with terminal, and which will so we'll open with terminal, because then we can clearly see the data set. Okay. And this is a terminal that again we have talked about this in another course, right, how to run these terminals and Linux inside the Windows. So, that is there in our earlier course. Okay. So, this is where we are. Okay. And here we have the data. Okay. So, in this class and subsequent class we will be using this data. So, the data we have is this raw counts file. Okay. This is what we have downloaded from the data, the GSC, right. So, that is the gene expression data. So, let us see this first. using the VI editor. Again, this is something we have discussed in our earlier course. So, here is the data. Okay. So, the first column is gene ID, from second column onwards these are the sample ID, right. So, these GSM numbers that we have seen in the NCBI GO. Okay. But it does not tell us, right, whether it is specific cancer type or normal tissue. So, those things we need the metadata, right, for to actually do that. The other thing we have is the gene ID. These are actually just simple ID, right, it is nothing else in here. Okay. So, how do you know the gene names and etc. So, we would like to get the gene name, because then we might be able to make some sense of this data, right. If we say the gene ID 729759, it does not make much, we cannot get any information, because it is of course impossible to remember all this, right. But if we look at gene name, right, like LDH, lactate dehydrogenase, we can make some connection, right, we know its function, etc. So, we need to also get the gene annotation. So, there are two things we need, as I mentioned in the presentation, is first we need the metadata, right, that will connect the sample ID to a specific type, right, whether it is a cancer, gastric cancer, diffuse gastric cancer, or intestinal gastric cancer, or normal tissue. So, this is something we will need the metadata for, and we also need the genome annotation, right, where we need this ID to common gene name mapping. Okay. So, we have both of them. So, the metadata, as I showed you, is the series matrix file. So, I have actually processed this series matrix process.txt. Okay. So, this contains this metadata information, and we can see this. Okay. So, here is the series matrix where we have the information. So, as you can see, here is the sample name, right, and this is the ID, GSM ID that we have, and it describes, right, which cancer type it is, whether it is intestinal type gastric cancer, or diffuse type gastric cancer, or normal mucosa. Okay. So, normal mucosa, this is the normal tissue, or whether we are getting different cancer types, right. So, we have this association file, and we can now include only certain samples, right, if we are interested in let us say diffuse gastric cancer versus normal mucosa, right, and we take these samples, some of these samples, and do the WGCNA analysis, so that we can identify some of the modules that are interesting, right, and we might be able to identify some of the modules that show different behavior in cancer versus normal tissue. Okay. So, this is the data set metadata, right, we have this information now. As I said, we also need the information, the annotation information, right, and this is the annotation information that we have. Okay. Where did we get this annotation information is from here. So, I will show you where we have this. Okay. So, it is not here. So, I have given the link in the presentation. So, perhaps you can have a look where we have that annotated information. Yeah. So, we have this annotation information in here. Okay. So, here is the annotation information, right. As you can see, here is the gene ID, right, these numbers that we have seen already, and here is the symbol, right, this is the common name of the genes, and it also has the description what is this gene, right. So, you can see much more details about this here. So, you have this information about name and then common name. So, these common symbols, some of them we can recognize, and we have the gene ID, not only the numbers. So, we have downloaded this information, and we also have this information with us here in this annotation file. Okay. So, again, just to read this file, right, the first column is gene ID, the second column is symbol, this is the common gene name, then you have some description what this gene is involved in. For example, you have, let us say, this is a dead H box helicase like 11 like 1, this is a pseudogene, right, like may not be functional, but you also have this all these for other genes as well, right. So, as you can see, for example, here you have a tubulin tyrosine ligase like 10 TTLL5, right. So, we can now get some information about the function of these genes, right, and it is easy to sometimes we can recognize some of these gene names. Okay. All right. So, we have the data, we have the metadata, and we have this annotation. Okay. With that, now we can actually start looking at the analysis part, right. So we can start with the first steps, right, and we can start loading the data, okay, and the R package, okay. All right, so we have seen this, right. So let's look into the WGCNA R code. For this analysis, what we will do is we will first choose the samples, right. So here are the samples that we are choosing, right, for diffuse type gastric cancer first, okay, and then for the rest we are for the normal we are choosing these samples, okay. So if you go back here, sorry, not here is the, okay. So here we have this data set, right, and from this data set, based on this annotation, based on the information that we have from the metadata, we will choosing these files, right. So based on these GSM IDs, so I'll just minimize this here, okay. So this GSM 3101062, right, this is the first one we have, right. We know this belongs to diffuse type gastric cancer, right, from the metadata. So we are including this 1062, 1063, and we have taken 9 such samples, okay. This is the diffuse type gastric cancer, and for normal mucosa we are choosing this GSM 3101068, 070, 072, 078, and so on, and again we have chosen 9 samples, okay. So it's not necessary you have to always choose 9, but we have chosen equal numbers, right. So you don't have to always choose equal numbers, you can have slightly different numbers as well, okay. So these are the samples that will actually process, and the first thing we have to also do, and that is something I have done, is actually use these numbers, right. So use the annotation file, okay. So we have the raw counts file, right, and using the, so as I said, in the raw count file we have the gene IDs as numbers. So I have used this annotation and I have converted that, and I have generated these annotated genes GSE 113255 diffuse gastric, okay, and these are the it also includes the normal samples, and we have the raw counts now, okay. And what I have done is I have, instead of the GSM IDs, I am using this T or N notation that is there in the data set, so that we can easily identify this, okay. So I have created this. So as I said, we are taking these 9 diffuse gastric cancer samples and 9 normal tissue samples, and so total 18. So what I have done is, from this original raw count data, I have taken those 18 and I have created a new data set, so which we can process with R. And then once we have chosen this, what I have also done is that I have used this gene annotation, right, so to actually change these numbers to common gene names, okay. So as you can see, I have changed these numbers now, so these are not numbers anymore, it's the common gene names, which could be useful for us when you go into the analysis, okay. So with that, as you can see here, we have about 39,375, right, values, rows, right. So we have a huge number of them, and we have 18, okay. So let's now go into R, and we'll start with the data loading part, okay. So let's move on to the right directory. Okay, so the right directory is okay. So we have moved here, and here we have the data, okay. So let's first load the data, and first load the library which is called the tidyverse, okay. Because we would like to work with the tidy format, so we are loading this library tidyverse. So I can show you. It's an R package. and it contains this tidyr package, dplyr package that we have talked about in earlier classes, right. So, so this is the tidyverse package here. So, you have a lot of this included already. So, if we get this tidyverse package loaded, and we can access all of these packages in here, right. So, for example, we have this table or the reader, tidyr, dplyr. So, we have discussed some of this in our earlier class. Okay, so here I have loaded this library, and it kind of shows us, okay, which packages are now attached. So, it also gives us some information about so by loading this package, we are actually masking some of the earlier functions. Okay, what does it mean? Because these packages share common functions. So this package is now overwriting or overloading. So they we cannot access those functions, right? So here are two things that are mentioned. For example, the dplyr filter, right? It masks flow core filter or stats filter. So stats is the base package. So all three packages, they have this filter function. So by using this by loading this package tidyverse, because we are loading dplyr, this filter function now takes over these other filter functions. Okay, this is something that happens, and as I said, if you still want to use this flow core filter function, the default filter function will be dplyr filter function. But if you still want to use the flow core filter function, what you have to do is you have to call something like flow core filter. Okay, right. Of course, we have to give the argument here, but this is how we will do, or you have to use stats filter. Okay. So, by default filter, you will get the dplyr filter function. Okay. Okay, so we have loaded this tidyverse. Now, what we will do is we will read the data, okay, and we will use this reader function, right, reader package in here. Yeah, so we will use this reader package here. So, okay, so again we have not talked about this reader package, but what it does, again you have this what kind of functions you can do, right. So it allows you to read this by something delimited files. Okay, so you have CSV you can read, right. You can say read CSV or read TSV. Okay, you can say read CSV2, which is semicolon separated values, or you can say read delimited files. You can have special characters. You can say this is the delimited special character that is there that you have to separate out. You can read tables, right. So we can do this, and we get tidy data. So that's very important, right. So we are loading this as tidy data. So then we can do a lot of these processes that we discussed, right. The data transformation organization would be much simpler for us. Okay, so we'll do this reader function, right. Read underscore delim, right. So we use delimited text, and we have to give this delim sign, okay, which is /t. Okay, we can use this read delim, or we can also say read TSV. So let's now use the reader function, okay, and let's give the file name, right. So this file name is the processed file that we have generated. Okay. Okay, so let's keep the full file name. Okay, so this is the full file name, and we have to give the delim, because we are calling the read delim function. So this is what we get. Okay. Okay. Okay, so let's see if what we have here. Okay, so it says 39,000 rows and 19 columns. Okay, so there is some warning message, but I guess this is fine, should not be an issue. We can also look at the data. Okay, so here is the data. So as we have loaded using this reader package, so this is a table. So it's a tidy data table. So we have again the dimensions are given, we have the gene IDs here in the first column, this is the character values, and we have the counts values, right. So these are the raw count values, okay, and we have many such like 39,000 such rows, and we have these values for all the samples. We have again 15 more variables it says. So we have these 9 cancer samples, diffuse cancer, and 9 normal samples, okay. All right, so this is what we have done. So we can also use this, right, in data, and we can also have, so it's, we can now see like what are the column names. So we have gene ID, we have the tumor samples first, 9 of them, and then we have the normal samples, right, 9 normal samples. You can see this N here, so that is the normal sample. Okay, so one of the things we'll do before we actually go into any of these analysis is do the drop NA values. Okay, so we have used this, remember I've showed you, right, we have this drop function, we can use drop NA or replace NA. So from the data we can drop NA, right. So we are dropping any NA value that's there in this data set, so we'll drop it. Okay. And let's see what we have here. Okay, head data, but let's say data. So the full data is 39,373 into 19. So actually there is almost none, right, only one, maybe one row was NA value. So that's good. So for everything we have NA values, right. So for everything we have some gene expression value, no NA value. Okay, so why we are dropping NA value? Because this might create a problem later on. Okay, so maybe that, so that's probably better, because all statistical analysis we might get NA at the end. Okay, so we have loaded the data and we have the NA, drop NA. So I'll just now illustrate the next steps. So before actually we move on to more complex ones. So the next step is actually doing something called data normalization. This is what we are going to do in the next step. So what we have done, right, we have looked into this data set, we have looked into the package, we have looked into the data set, and then we have actually prepared the data. We have seen the sample metadata, we have done the gene annotation, and I have combined all of them together, and we have created a process data file which I have loaded, and we have done the first step, which is the drop NA. So now I'll go back because I want to show you some of these WGCNA functions which we did not check very carefully, right. So one of the things, first thing we'll have to do is called normalization, which is actually not here, but then what we'll use is something called network function. So let's see, I'll show you where this network function is. Okay, so we have this network function. So we have to use this peak soft threshold function. We have to use the network function. So what I'll do is I'll actually blockwise modules. So I'll show you these functions. So one is blockwise modules here. So this is something we'll use in the next subsequent class. Okay, so sorry. So maybe it's let's see. Yeah, so here it is, blockwise modules, page 55, and this function is actually useful for automatic network construction and module detection. So this is the function that we'll use to do the network construction and for the module detection. And these are some of the parameters that are in there. For example, we have to give such as this max block size penalty, power, etc. So what should be the block size maximum that is allowed in our construction process? What is the correlation type? Whether you should use Pearson correlation or Spearman. There are a lot of these functions, and they are actually described below, right, as you can see. So what are the blocks, right? These are the blocks in hierarchical clustering and modules, right? What is the max block size that we can use? Correlation type, right? All these things are described. the power, soft thresholding power for network construction. As I said, we need to pick a soft threshold. So, that will actually be used for this module network construction. Okay. What kind of network type we should create, whether it is unsigned or signed network. So, all these things are in here. So, all these details are in here. The other function that we want to use is the pick soft threshold. Okay. So, here we actually want to create this scale free topology, right. So, as I said, we want to get a scale free network, and in the next class I will describe what is scale free network. So, this is something we want to do. So, for that we need to pick soft threshold, and this is the function pick soft threshold, and here are again the parameters. There is a large number of parameters, they are again described how each of them would work, right, and we will use this in the next class. Okay, so, what we get is this power estimate, fit indices, etc., that we get here, and then we can use this soft thresholding power to actually generate the network. Okay, so, we will discuss about this in the next class in much more detail. So, we can go back to the presentation. So, here are the references for this class. So, what we have done in this class, we have had an overview of the R package WGCNA, right. We have looked into some of the functions that are there, and we have also introduced the data that we are going to use in the hands on, right, for this class, starting with this class and for the next few classes. So, this data set is a gene expression data set which contains gastric cancer samples as well as normal samples. So, what we have chosen are 9 gastric cancer samples and 9 normal samples. We have put them together along with the gene annotation, right, name of the genes, and we have now processed that file, and we have loaded that file in R. And what we will do is we will then in the next class we will start with the data and we will go through each of the steps. First, I will introduce what are the steps and why we are doing each of these steps. Followed by that, we will do the hands on, and we will actually execute those steps with our package WGCNA. So, with that I will close this class. Thank you.